Hello and welcome to this video lesson on the National 5 biology topic, distribution of organisms. We're on Unit 3, Life on Earth, and this is Key Area 2. Once again, here's the SQA core specification or mandatory knowledge section for this topic. Remember, this is all the bits of knowledge and content that you could be tested on in an exam or a test at this level for this topic. Again, this might look like a short topic at first, but there are quite a lot of new definitions and terms, along with some new techniques that you need to learn about too. As usual, as we go through the theory for each section, there'll be some questions after each part so that you can be sure you're ready to move on. So our learning intention for this part is to learn about distribution of organisms, and hopefully by the end of this lesson, you'll be able to describe abiotic and biotic factors and give examples of each, explain how to measure abiotic factors along with possible sources of error and how to minimise them, explain how to sample plants and animals and evaluate limitations and sources of error in their use and construct paired statement keys, describe the effect of biotic and abiotic factors on biodiversity and the distribution of organisms, and finally explain what an indicator species is. So our first section of this topic is going to be on abiotic and biotic factors. So the biodiversity and distribution of organisms, so where they're found within an ecosystem, is due to both abiotic and biotic factors. Organisms will only want to live in areas where the biotic and abiotic factors suit them. So for example, they might need to have appropriate levels of light intensity and temperature with low levels of competition to be able to survive and thrive in that area. First, let's have a look at abiotic factors. So abiotic factors are what we call non-living factors. Now, the way to remember this is bio means living, biology, our topic. A means non, so non-living, so non-living factors. And examples of these include light intensity, moisture, pH, and temperature. Now, all of these things are non-living factors. Light intensity, moisture, pH, and temperature are not affected by living things. One thing to point out here is you cannot just say light by itself. You have to say light intensity. So all of these are non-living factors, but they will still have an effect on where organisms are distributed within an ecosystem. Biotic factors, on the other hand, are living factors. Now, there's quite a few examples for this one, and you do need to know all 5. So you have to be able to pick these out from examples in the future in some past paper questions. So examples include competition for resources, disease, food availability, grazing, and predation. So if you think about it, all of these things are to do with living things. So they are living factors, which mean they are biotic factors. So what we're going to do now is we're going to try some quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. Okay, so for question number 1, it asks to give 3 examples of biotic factors. So they would be competition for resources, disease, food availability, grazing, and predation. Number 2 asks to give 3 examples of abiotic factors. So they're non-living things. So light intensity, moisture, pH, and temperature. And remember again, you have to say intensity after light. These are 2 past paper questions that I picked out to show you. So which of the following statements is true of predation? So there's 2 things this question is asking. We can see that from the multiple choice options we have here. The first 2 say it's an abiotic factor and the second 2 say it's biotic. So that's the first thing we need to choose. Well, we know predation is to do with living things, so it is a biotic factor. So this means we can cut it down or narrow it down to C or D. Now, the next thing it asks is, does this cause a decrease or an increase in prey numbers? Now, remember we talked about predation in our last topic, and we talked about what predators and prey are. So if there's a lot of predation, is that going to lower the prey numbers or is it going to increase the prey numbers? Well, it's going to decrease the prey numbers because more predators means less prey. So it is C is the final answer for this one. An example of a biotic factor affecting a population of plants is, so again, we need to think, what does this mean? So biotic is a living factor, okay? So let's run through these. So a leaf disease reducing the growth of lettuce plants. Now, disease was one of the biotic factors that we were talking about before, so I think this one sounds right, but I'm going to check my other 3 first. Acidic soil preventing the growth of daisies. Well, this is pH, and we said that pH was an abiotic factor, so it can't be B. Shade from buildings causing a decrease in the growth of grass. Again, that's to do with light intensity, and that was one of the abiotic factors, so it can't be the answer to this one because this is about a biotic factor. Finally, a cold winter causing a decrease in the growth of geranium plants. Cold winter decrease means temperature, and temperature was another one of those abiotic factors. So that means that disease is the only biotic factor here, so A is our correct answer. Okay, now we're going to move on and look at how we measure abiotic factors first, and then we're going to look at biotic factors after. So first of all, you must know how each abiotic factor is measured. You also have to know for each one possible sources of error and how to minimise them as well. Now, this piece of equipment on the right-hand side, you'll get to try out in class. So it has a light sensor, a pH probe, and a moisture probe, and we're going to come back to all three of these in a second. But this is just what it looks like so that you can picture it as we go through and we talk about how to use this piece of equipment. So the first thing we're going to look at is light intensity and how to measure that abiotic factor. So how you measure it is you use something called a light meter. Now the light meter was on that one we saw before, but here's a light meter here as well so that you can see it. Essentially, you take this meter and you hold it at the soil surface and you point it in the direction of maximum light intensity. So basically towards the sun or towards whatever the light source is. This will allow you to see how much light or the light intensity that plant or those plants are being exposed to. Now, for each of these factors, apart from how it's measured, you also have to know possible sources of error and how to minimise them. So a possible source of error for this is standing in the path of the light. So if I was holding this light meter down near the ground, but I was standing with my back to the sun, I might cast a shadow over the light meter. And if I did that, that would give an inaccurate reading. So what I need to make sure I do, because that's a possible source of error, I would minimise this by being careful where I'm standing so I don't cast a shadow onto the light meter. So be very careful. Some questions will ask you for the possible source of error, and some will ask you specifically how to minimise, and you don't want to get these two mixed up. Our second abiotic factor that we're going to look at how we measure is temperature. So to measure temperature, there'd be no point in using a light meter that we just spoke about. So temperature of the soil can be measured by using a thermometer. So what we do is we place it in the experimental area, we allow it to stabilise, and then a reading is then taken from that scale. Now here there are two possible sources of error that you can talk about. You can either talk about the fact that there might be leftover soil on the probe after you've done a reading. So if you put the thermometer probe into the ground and then take it out, there could be some soil still stuck to the outside. And then if you go and put that into another bit of ground, that leftover soil could give you an accurate reading again on for the temperature. So one way to minimise this possible source of error is to wipe down the probe after each use or between samples. That would mean there'd be no soil left over on the outside, and then you would get the right temperature reading. Another possible source of error is taking the reading too quickly, and again that can give an inaccurate reading. You want to keep saying inaccurate reading in your possible sources of error. Now, this will happen because you're not giving it time to adjust. So the way to minimise it is to leave it for a time to adjust before taking the reading. So essentially put the thermometer into the soil, wait for a certain period of time, and then take the reading. The next two abiotic factors, so pH and moisture, we're actually going to talk about together because they're measured using a similar probe or meter, and they have the same sources of error and ways to minimise them. So we showed you this at the start. So we had the light sensor, which we've already talked about, but we have a moisture probe and a pH probe, and they're both actually usually, now you get them attached to the same meter. So if we want to measure soil pH, we use the pH probe or pH meter. If I want to measure soil moisture, I measure it using a moisture meter. In both cases, this probe is pushed into the ground and a reading is taken from the scale on the meter. Now, a possible source of error here is that leftover soil again can be on the probe and give an inaccurate reading, just like on our last example. And again, my way to minimise that would be to wipe down the probe after each use or between samples. Again, like before, you can also take a reading too quickly and that can give an inaccurate reading, so leave time for it to adjust before reading. And again, don't get these two mixed up. Make sure if the question asks for a possible source of error, you're giving this and you're not just giving the way to minimise it. Okay, so let's try some more quick questions on what we've covered so far to check our knowledge before we move on. So once again, pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. Okay, so question number 1 asks you to name the instrument used to measure light intensity. That's a light meter. Number 2 asks to name the instrument used to measure soil moisture, and that's a moisture meter. Number 3 asks you to describe one source of error when using a light meter. So that would be standing in the path of the light, which can shade the meter and can give an inaccurate reading. So remember to try and put that inaccurate reading bit in there. Number 4 asks you how could that source of error be minimised? By being careful where you're standing so you don't cast a shadow on the light meter. Number 5 asks you to describe one source of error when using a soil pH meter. So one thing you could say was that leftover soil on the probe could give an inaccurate reading. Or for number 2, you could say that taking the reading too quickly would be an error, and it might give an inaccurate reading again. How could these sources of error be minimised? So whichever one you chose, if you chose number 1 about the leftover soil on the probe giving an inaccurate reading, you would say you would wipe the probe down after each use or between samples. But if you chose number 2 about taking the reading too quickly, you could say you leave time for it to adjust before reading. Okay, so now what we're going to talk about, we've talked about how to measure abiotic factors in an environment. Now we're going to talk about sampling plants and animals themselves. So it is impossible to count all of the plants or animals in a habitat. You're not going to go out and be able to count every single plant, every single blade of grass, every single animal, bird, insect in that area. So what we do is we take a sample of them and we use this to estimate the abundance, okay, so the total number of them. So you can estimate how many animals and plants are in an area using various techniques, but you have to choose the technique which best matches that organism, okay, so whether it's a plant or animal that you're trying to sample. So if you want to estimate the abundance of plants or slow-moving animals, you can use something called a quadrat. For sampling invertebrates, which live on the ground, you would use a pitfall trap. There are obviously things that are bigger than small invertebrates that live on the ground. There are things that obviously you've got birds that fly, you have larger vertebrates, but we're not going to focus on those in this topic. So we're just going to look at quadrats and pitfall traps. Again, for both of these, you have to know how they're used and sources of error in their use as well. So we're going to start with quadrats. So quadrats are used to estimate the abundance of plants or slow-moving animals. Now the animals have to be slow-moving because once you throw your quadrat and you try and count them, you don't want them to be moving box or leaving the quadrat behind. Now how you use this, the quadrat is this thing down here. This is thrown at random and the number of squares that contain the investigated organism are counted. So in this example here, maybe I was going to count clover. Well, I can see there's some clover in these boxes, so I would count this box, this one, this one, this one, this one, and this one. This box over here and this one over here don't have any clovers at all in them, and this one we'd have to decide about because it actually only has part clovers that are already in another box, but I'll explain that in a wee second as well. So to get a representative sample, okay, of the total population of the organism that we're trying to count, you have to do an adequate number of quadrats and they have to be randomly thrown. An average number of each organism per quadrat is calculated and then scaled up to represent the area. So what I mean by this is, let's imagine that I have a field. So my field is 50 metres by 20 metres. Really large field, and let's imagine, this is not to scale, but this is my quadrat. Now, doing one quadrat is not going to be able to tell me or let me scale up and find out the abundance of plants in this field. To get a more representative sample, I would need to take multiple quadrats. Now, in this example here, I've done 5. However, the more that I can do, the better, and the more representative that sample will be of the actual population. So I have done 5 randomly placed quadrats on this field, but now I want to work out what the actual estimated abundance is of what I'm looking for. So here I'm focusing on daisies. So I want to know an estimate of how many daisies are in this field. So I've randomly thrown my 5 quadrats, and what I can say is, imagine that each quadrat only covers an area of 1 metre squared. So let's imagine my quadrat is 1 metre by 1 metre. First thing I need to do is I have to work out the area of the field. So my field was 50 metres by 20 metres. So I do 50 times 20, and that gives me 1,000 metres squared, which is the basically square footage of my field. If there is an average of 4 daisies per quadrat, so let's imagine, I've not put the data here, but let's imagine there's an average of 4. So I've got my numbers from my 5 different quadrats, how many daisies there are. I've added them together, I've divided by 5 to get the average, and it's 4. So to work out the estimated abundance of daisies in the whole field, not just in one quadrat, I would times the 4 by 1,000, so the metres squared, and that'll tell me there's roughly 4,000 daisies is my estimated abundance. So this type of question doesn't come up very often, but if it does, that's the way you work it out. Okay, now let's talk about the limitations of a quadrat. So a quadrat can only be used for stationary or slow-moving organisms. This is not going to work for large organisms, and it's not going to work for ones that will move quite quickly away if you try and throw a quadrat on top of them. Second limitation that quadrats have is it depends largely as well on human accuracy too. The main thing you need to know, though, is you need to know about the sources of error with a quadrat, just like we did with our pH, moisture meters, temperature meters, thermometers, and light meters as well. So the largest source of error that we normally have with a quadrat is called non-random sampling. So inaccurate results can be made if you choose where to throw. So for example, if I walk into a field and there is some daisies, lots and lots of daisies down one end, and the other end of the field doesn't have very many. If I walk into my field and choose the bit that has lots of daisies and just place my quadrat down there, that is not a random sample, and that is a big source of error. If I choose those places that have the most daisies, when I finally work out my total abundance, there's going to be way too many compared to what there actually is, because I've ignored that other part of the field where there wasn't any. So the best way to take out this bias from humans is to make sure you randomly sample. So you randomly choose where to place the quadrats. And you can see this if I go back, because some of these ones are actually quite close together. If I was choosing where to put these, I'd probably space them out, but that's not a good thing to do. So I randomly thrown these quadrats, and this is where they've ended up. And this will give me a more representative sample. But if I did non-random sampling, that would be a source of error. Now, the way to minimize that is just to make sure you place the quadrats randomly. Another source of error, but I wouldn't go for this one if you have the option. If you have the option, I would talk about this one. But another source of error is whether an individual organism is in a certain square or not, so whether you count it or not. So for example, an organism on the outer edge of the quadrat, do you count that or do you not count it? So again, if I go back and I show you here, we have here in this picture, there's a dandelion that's right on the edge there. So essentially, do I count that in this box or do I not? So essentially with quadrats, you only count the box once if there's even 5 things in it. So even if there was 5 dandelions in this box, I would still just count 1 out of 25 or whatever the number of squares is. That's a big source of error. Do you count it? Do you not? So a way to minimize this is to set it up and follow certain rules when using the quadrats. So some scientists decide borderline organisms, so those ones that are on basically the borderline of the quadrat, if they're found on 2 specified sides, they might count as in, whereas maybe on the other 2 sides, they count them as out. Now, that's just an example. You don't need to know this. As I said, go for this one for sources of error if you can, but this one here is just another example of what you could say. Okay, so we've talked about pitfall traps, which are used to measure or estimate the abundance of plants and slow-moving organisms. Now we're going to look at pitfall traps. So pitfall traps are used to sample invertebrates which live on the ground, and that's really important. This is not about invertebrates that live up higher, it's not about ones that can fly, it's only organisms that walk along the ground and they're small enough, so it's invertebrates that get caught in these. So how you use it? So essentially you dig a hole in the ground and use a plastic pot and you place it into that hole in the ground here, with the top of the pot level with the ground to ensure that the vertebrates will fall in. So invertebrate comes along and falls into the pot and gets trapped. The trap is then camouflaged and left for a period of time, after which the number of each invertebrate found within the trap is counted. So you set this up, you leave it for as many organisms as possible to come along and fall in, and then after, say, 24 hours, you come back and you check the trap and you count how many are there. Now, there are some limitations to pitfall traps. This can only be used to sample small invertebrates that are found in the ground and can't fly. Pitfall trap samples may also not be fully representative of the area, so to overcome this again, just like Quadrats, scientists would do several traps and would calculate an average from each of them. Once again, we have to know the sources of error and how to minimise them. So potential sources of error, for this there's quite a few. So the first one would be water could get in when it rains. Now the way to solve this is by putting a few pinpricks in the bottom of the plastic cup, and that'll allow the water to drain out of the bottom. Second potential source of error is the birds taking or eating the trapped animals. So if birds manage to find this is like a little cup of all its food for it. So what we want to do is make sure we hide or camouflage the trap. Now this is a really important one because this is the most common one that you'll probably say, but you have to be careful how you word it. You're not allowed to use the term lid because if you say that you put a lid on top of it, that implies that you have sealed the container. So if I go back to this, if I put a lid on this cup, nothing is going to fall in. So I'm not actually going to trap anything. So you don't want to say the word lid, you want to say you're going to camouflage or hide the trap. The third potential source of error is that predatory animals like spiders, ground beetles, or centipedes might actually eat the other captives that are in the trap. So if they're left too long, and I have some various little invertebrates in here, but then say a spider comes in, so falls into my trap, it might actually eat all of the other ones, and then I'll just find the spider when I come and look at the trap. So that's only if it's left too long. So the way to minimize that is to check the trap more often, so it's shorter intervals. Okay, so let's try some more quick questions on what we've covered so far to check our knowledge before we move on. So pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we will go through the answers. Okay, so the first question asked, a student wants to investigate the biodiversity of small invertebrates living in an area of grassland. Name a sampling technique they could have used. So pitfall trap would be our answer for that one. Number 2 asks, how do you ensure using a quadrat that you have a representative sample? So you'd throw the quadrat many times randomly, and then you would take an average of the results. Describe one source of error when using a quadrat. So inaccurate results can be made by choosing where to throw. And how do you minimise that source of error? Well, you make sure you place the quadrats randomly. Number 5 asked to describe one source of error when using a pitfall trap. Remember, we had lots of options here. So you could have said about water getting in when it rains, birds taking or eating the trapped animals, or predatory animals like spiders, ground beetles, or centipedes that are eating their fellow captives. You don't have to give all of these examples. How can those sources of error be minimised for number 6? Well, if it was number 1, so the water getting in, you would put a few pinpricks in the bottom of the plastic cup to allow it to drain. If it was number 2, you would hide or camouflage the trap. And if it was number 3, you would check the trap more often. Okay, so now we have looked at abiotic and biotic factors in more detail, and we've looked at what the different types are. We've looked at how you would measure abiotic factors, and then we've looked at how we'd sample animals and plants. Now we're going to look at how we identify those organisms that we sample. So if I'm looking at my quadrat, or I'm looking at my pitfall trap contents, and I need to work out what those organisms actually are that are in them, because apart from spiders and stuff, it might not actually be as easy as you think to identify what that organism is. So we can use something called a paired statement key. So biological keys are used to identify organisms. A paired statement key is a list of statements which describe certain observable features of an organism and then let you basically figure out which one it is. So here's an example of a paired statement key. So we have various statements that are made up of 2 parts. So there's 2 statements for each question, and you basically look at it, they're usually the opposite of each other. So leaf made up of 1 part, or leaf made up of several smaller parts, or outer leaf edge is wavy, outer leaf edge is smooth. So they're very, very similar to each other, okay? And this allows you to split up different organisms or different organisms into groups. And essentially, they will have instructions at the end of each statement, which tells you to either go to another question or it gives you an answer of what you're looking at. So if we look at this plant down on the right-hand side, this leaf here, best thing to do is pause the video and see if you can work your way through from question 1 and see if you can figure out what the name of this is. Okay. So this is a horse chestnut leaf. So to identify this one from pair number 1, the statement which applies is the leaf made up of several smaller leaflets. Therefore, we have to then go to number 3. So it says go to 3. So then I go to number 3, I can skip number 2 because it's told me to do that. And then for pair number 3, the statement which applies is leaflets attached at a single point on the leaf stalk. So therefore, my answer is horse chestnut. Let's try another one. So pause the video here, use this same paired statement key to work out what this leaf is. Okay, so this one is a laurel leaf. So again, to identify leaf B from pair 1, the statement which applies is the leaf is made up of one part. So this time I go to question number 2. When I go to question number 2, for pair number 2, the statement which applies is the outer leaf edge is smooth, so it tells me to go to question number 4. I go to question number 4, and for this one, the statement which applies is the leaf is longer than it is wide, so that means it is a laurel leaf. Okay, so let's pause here and try some quick questions again on what we've just covered. Just a really, really quick one that is on paired statement keys, just to check you've understood it. Now, you can be asked, apart from identifying a particular picture and trying to use a paired statement key to do it, you can be asked to actually construct one yourself. Now, normally what they'll do in a past paper instead is they'll give you a mostly constructed one, but they'll have left some blank spaces. So pause the video here and using this information in the table, try and fill in A, B, C, D, E, and F on this paired statement key. Okay, let's run through our answers. So with this one, it's a lot of problem solving. You're trying to kind of work back the way almost. You can already score out brimstone, common blue, and small heath because they're ones that we've got answers for already. So basically near the end here, you're expecting your other two to come up. You can follow through and make sure you can see what things they could be split up by. So your answer for this one should have been, for A, it should have been 2. For B, it should have been 3. For C, it should have been eye spots present. For D, it should be eye spots absent. For E, it should have been small white. And for F, it should have been gatekeeper. If you have any questions about that, I would go back and have a wee look again at that table and compare it now to the answers and see if you can work it out. Okay, the last thing we need to learn about in this topic is indicator species. Now, this is a really important definition that you need to know off by heart. So indicator species are species that by their presence or absence indicate environmental quality or levels of pollution in an area. So the main thing here to try and look for, if you're looking at a definition and you're wondering what the term is, if you see presence or absence, then that means it's an indicator species definition. Now we're going to look at 2 examples. You don't need to know both of these, but this is just to try and highlight what this definition means. So lichen are indicator species of air pollution, and many freshwater invertebrates over here are indicator species for water pollution. So we're going to look at both of these in turn. So lichen are used as a biological indicator of air pollution. So if you want to know how polluted air is in a certain area, you can look for these lichen on the trees. So they look like this. Now, different species of lichen can be found depending on the levels of sulfur dioxide or pollution that's in the air. So if we have a look here, if there's a very low level of pollution, you'll find on the trees hairy, shrubby, leafy, and crusty lichen. As you move down with increasing air pollution, you'll see the biodiversity actually decreases, and in high levels of pollution, areas where there's high air pollution, you'll only see crusty lichen on the trees. This is why it's important that we talk about presence or absence, because presence, well, there's only one that's present here, and this is present all the time. So if we just saw a crusty lichen, we wouldn't be able to tell which level of pollution it was. However, because we are missing the leafy, shrubby, and hairy from this one, I can tell that it's a high area of high air pollution at this point. If I look around the trees and I can see all 4 of my species of lichen, then I know that the pollution level is really low. So presence or absence of these indicator species will tell us about the environmental quality or the level of pollution in those areas. Now, lichen aren't found underwater. So with water, you can use freshwater invertebrates as a biological indicator of water pollution. So rivers with a low oxygen concentration contain species such as sludge worms, which can survive in low oxygen conditions and high levels of pollution. So what you find is because, basically in more polluted areas of river, you'll find that they have a lower oxygen concentration, and in clean bits of the river, they'll have a higher oxygen concentration. So at the areas where you can see here, there's untreated sewage being added into this river, the clean river came before it, and then in this really highly polluted bit of river, you see that we have these things called sludge worms. So if you find a sludge worm, it means it's quite a polluted bit of water. Then as it goes down this river, obviously the river will keep moving, it'll get diluted with more water, and basically it'll get less pollution, basically lower concentration of pollution in it at this level. So then you start to see things like water louse, which live in a kind of medium level of pollution in water, and then you get mayfly nymphs again, which are found in clean river. So at the end and at the start, we had our clean river, and that's where we found mayfly nymphs. So they're an indicator species of it being clean river, not polluted, whereas sludge worms were an indicator of it being polluted. So as I said here, small invertebrates live in freshwater, have particular levels of pollution that they can handle. By knowing what the tolerance of particular organisms are and looking at what you find, you can decide on what the quality of the water is. So here again, we have increasing levels of pollution going this way. So if I look at a bit of river and I take a sample and I find that it has leeches, bloodworms and rat-tailed maggots, then I know that area of river or that bit of water is basically polluted. If instead I find in my sample that I have stonefly nymphs and flattened mayfly nymphs, that would mean that it would be a low level of pollution. And by not finding bloodworms, leeches and rat-tailed maggots, again that highlights that this is an area of low pollution. So again, by their presence or absence, you can use indicator species to tell you about the level of pollution or environmental quality of an area. Okay, let's try a final set of quick questions on what we've covered so far to check our knowledge before we finish up. So pause the video here and try these questions, either by saying the answer out loud or writing them down. And when you're ready, play the video and we'll go through the answers. Okay, so question number 1 asked, the biodiversity of key insects can reveal information about the environmental quality of an ecosystem. What name is given to the organisms which by their presence or absence indicate the levels of pollution? So presence or absence we know as indicator species. The following key can be used to identify invertebrates found in fresh water. Use the key to identify the following 2 invertebrates. So if we look at this picture here, it says legs present or legs not present. Well, it's definitely got legs present, so I'm going to go to number 2. 8 legs or more than 8 legs? Well, 1, 2, 3, 4, 5, 6, 7, 8. So 8 legs, which means this is a water mite. Looking at this one and starting from number 1 again, legs present or legs not present? Well, they're not present, so I'm going to go to 3. Shell present or no shell present? There's no shell in this picture, so I'm going to go to number 4. Segmented body or non-segmented body? Well, this is all just one big segment, so non-segmented body, which means that this is a flatworm. Okay, so that's us finished learning about distribution of organisms. I hope that you're now confident about describing abiotic and biotic factors and giving examples of each. I hope you can now explain how to measure abiotic factors along with the possible sources of error and how to minimise them, and remember to keep those separate. Explain how to sample plants and animals, so using quadrats and pitfall traps, and you now know about their limitations and their sources of error in their use. I hope now with that last question especially, you can now use and construct paired statement keys. Hope you can describe the effect of biotic and abiotic factors on biodiversity and the distribution of organisms. And finally, you should now know your explanation or definition of what an indicator species is. Now, as I said, this is quite a difficult topic with a lot of new information, definitions and techniques, so please feel free to go back and watch parts of the video again in the future if you need a refresher on any of the parts of this topic. Thanks again for listening.